Good afternoon, everybody, and thank you for joining us to celebrate the Day of the Immunologist from the Asociación Colombiana de Inmunología, ACOI. It is really my pleasure to host this session. My name is Manuel Franco, for those of you who don't know me, and I am the president of the Asociación Colombiana de Inmunología of ACOI. And our session today will be focused on single-cell omics. It is my conviction that the methods, the techniques, the strategies, the instruments we use for our science determine how science is made. And right now with single-cell omics, I think immunology is being transformed completely. And for this purpose, we have invited two groups of people. We have invited Dr. Benjamin Mekif from the Vijayanand Lab in the Hoya Institute for Immunology. He's a postdoc in this lab who has been using these methods of multi-omics, single-cell multi-omics to study SARS-CoV CD4 T cells. And I think that this has given a really next step understanding of what these cells are and what they're doing, and and give us a vision of what they're doing. And in the second phase of our talk, we will have talks by Adriana Suarez and Anja Bastian from 10x Genomics. It is also my conviction that interaction with the companies that provide us with all these reagents and instruments is key. And 10x Genomics is really an example of this type of company. And you will hear from them in the second part of our talk. So without any more introduction, I will give Dr. Mekif the floor so he can start his talk. Dr. Mekif, please.
Thank you very much. And thank you very much for that introduction. So today, I'm going to talk to you about some of the work that we've done over the past year or so looking at SARS-CoV-2 reactive CD4 T cells. And this is principally using single-cell sequencing technologies. And all that I'm going to talk about today has been done using the 10x platform. I'm not going to go into a huge amount of detail about the platform itself because I'll leave that to the experts to kind of describe it in a much better way than I could. But yeah, so I'll take you through some of the stuff that we've done recently. So I'm based at the La Jolla Institute for Immunology here in San Diego. I'm supervised by two people. So Christian Ottensmeyer, who's an oncologist based, who was at the University of Southampton. He's now at University of Liverpool. And obviously Professor Vijayanand, who is the PI here in San Diego. So I kind of broken down my talk into a couple of different sections. So I'm first going to describe the assay that we use to pick out our viral reactive cells. I think it's important to kind of go through a little bit of the history of that and why we've chosen to use that as our kind of baseline and what we use to actually get antigen-specific cells. And then I'm going to talk about some of the single-cell RNA transcriptomics that we got with our SARS-CoV-2 reactive CD4 T cells from COVID-19 patients, some of the TCR analysis, and then I'll briefly touch upon some of the memory SARS-CoV-2 analysis that we've done recently. I won't go into great detail because this data isn't published, but there is some things that I think might be interesting just to mention briefly. Is it going to let me go forward? So we've used what we call the RT assay, so it's antigen reactive T cell enrichment assay. Now, the idea behind this is that you isolate cells that upregulate expression of CD40 ligand, CD154. Sorry, I'm having to let people in as I go. CD40 ligand upregulated after peptide stimulation. So it was back in 2005 that two papers in Nature Medicine showed that actually you could stimulate cells with peptides. Then after 6 hours, you could get nice upregulation of CD40 ligand. There were two groups that published similar work at the same time. one showing a nice kind of time course, and another one which showed that actually you need to put CD40 blocking antibody, because the interaction that you have between a T cell and an antigen presenting cell, as soon as there is that interaction between CD40 and CD40 ligand, the T cell will down-regulate CD40 ligand. So you need to block that interaction in order to maintain expression of this protein on the cell surface. So these two groups kind of, first showed this in 2005, and Alexander Sheffield, who was on this second paper, was one of the people who really kind of moved it forward and did some more validation. So in 2014, they kind of showed that actually what you'd want to do is do some column enrichment. You want to enrich these CD154 positive T cells, because when you do that, you get a much, much better signal to noise ratio. So instead of having this kind of plot here that you have here with a few dots kind of in the CD154 TNF alpha double positive, if you enrich, you get a much better signal, and the background noise is much, much lesser. So, as this plot shows here, doing a column enrichment means that you're not kind of unsure about where to draw the gating lines. It separates out a lot nicer. And then a few years after that, this is the same group right the way through, that's also published two very, very nice cell papers, which I would recommend reading. One of them looking at regulatory T cell directing tolerance in neuroantigens in humans, and then looking at Th17 immunity and how that relies on cross-reactivity with Candida albicans. But this first paper here on the left, what they showed is that actually using this assay, you can isolate two different populations. You can isolate effector populations by looking at upregulation of CD154, but you can also isolate regulatory T cell populations by isolating cells that upregulate expression of CD137, which is 4-1BB ligand. And what they showed very, very nicely is, if you look at these kind of cells in red here, after 6 hours of stimulation, if you pick out or you isolate cells that have only upregulated CD137, they display all the classical markers that you want of regulatory T cells. So FOXP3, CD25 high, CD127 low, CTLA4. So it really shows that within 6 hours, if you have this kind of these separate populations of 154 for effector cells and CD137 for regulatory T cells. And so we've built upon all this work that's been done by Petra Backer and Alexander Sheffield in Germany. And following on from that, last year, we published a paper, or the lab published a paper showing that combining this RT assay with single cell transcriptomics. And this was looking at allergen specific T cells. And as you can see from this plot here on the bottom, we did kind of the same isolation. So we isolated CD154 enriched cells for kind of T helper effector populations, and then CD137 high, CD154 negative for regulatory T cells. And you can see here from some of the kind of gene expression that we have, lots of genes associated with activation. And then you can, the two separate populations that were isolated can, separate out when we do a t-SNE plot between the kind of the CD137 and then the CD154. And then if we look at gene set enrichment, so taking some genes that are associated with a regulatory T cell signature, we can show that actually these cells isolated using CD137 are enriched for a regulatory T cell signature. So that was kind of where we'd combined the assay and the use of 10x genomics to do single cell RNA sequencing. So before the pandemic, I joined the lab in, I think it was June 2019. And the idea for me was to look at phenotype and functional profiles of pathogen and tissue specific CD4 T cells using single cell RNA sequencing. So kind of move, going a step further from the allergen specific T cells. And this was going to be a very simple project. We were going to take PBMCs from healthy people, healthy lung tissue and lung lavages, and we were going to stimulate them with peptide pools to very common viruses like EBV, CMV, and flu. And so I'd done this with a very small subset of donors, and we managed to get a kind of approximately 10,000 cell single cell data set. And from that, we were already able to see that actually between the three viruses, there were quite distinct kind of transcriptional profiles in the viral reactivity, which was, kind of I guess as immunologists, we like to stick to the things we know, and viral reactive T cells, people will mostly tell you they make interferon gamma. That's how you know they're a Th1. But this was kind of already showing that actually you could split them out into different subsets. There were kind of different gene expression profiles in the 3 viral reactivities. So that's where we'd got to in early 2020. But then obviously, as with everyone else, priorities shifted and we moved into doing COVID work. So at the time, we were already setting up a study with the University of Southampton in the UK. And what we were going to do is we were going to take blood and tissue samples from people with known viral infections. And we were going to be able to track them over time and perform single-cell RNA sequencing, look at what's happening in the circulation, look at what's happening at the site of infection, and kind of, track that over time to see what happens. So we already had the kind of the methods in place, if you will, to work on antigen reactive SARS-CoV-2 specific CD4 T cells. We had the technology, we had the analysis that many people in the lab had done before. So we were ideally positioned to kind of do this, translate this work to SARS-CoV-2, which is what we did. And we spent I call it an intense couple of months to try and get this work done, because everything, in that time frame was time sensitive, and as I'm sure you're aware, competition was fierce. So we had an intense few weeks to kind of get the data, analyze the data, which was done in no short part by Ciro and Vicente, who's here today. And then we eventually got this paper published in Cell, looking at SARS-CoV-2 reactive CD4 T cells in COVID-19 using single-cell transcriptomics. So this is kind of just the basic plots that to show what our kind of sorting profile looks like. This is what the cells look like when you take them off, once they come off the enrichment column. So we have a very, very clear population, double expressing CD154 and CD69. So there's no kind of ambiguity as to where you draw the line. The populations are well defined, and we get very, very nice staining from that. And ultimately, we ended up with over 100,000 single cell transcriptome for virus-specific cells. Now we had COVID donors as well as some healthy donors to use as sort of controls. So we had we had 2 kind of categories to kind of compare what the viral reactive CD4 T cell transcriptome looked like. So for the COVID-19 patients, they were collected between 2 and 47 days from symptom onset. So it's quite a wide range, but I think that's kind of fairly common. Some people, presented with disease very quickly. Some people, it took a little longer. And the days in hospital for our hospitalized cohort range from 4 to 46 days. We had some people who were in ICU and some people who were non-ICU. We didn't differentiate between the two, because there are so many sort of cofactors that could lead to a donor being put into ICU compared to non-ICU. So we just grouped them together and had them as hospitalized and non-hospitalized patients. We also had blood from healthy donors that were collected before the pandemic. So these were cells from people who'd never seen the virus. And we also had a cohort of donors who, younger donors who we tested for influenza reactivity. So looking at the kind of SARS-CoV-2 specific cells, we did generally find that we could pick up more SARS-CoV-2 reactive CD4 T cells per million PBMCs in hospitalized patients. These cells were stimulated with overlapping peptide pools directed towards the membrane protein and towards the spike protein. So those are the 2 proteins that we focused on. Those were peptide pools that we got from Miltenyi that we found to be pretty reliable. So we use those throughout. Whenever I talk about SARS-CoV-2 reactive CD4 T cells, that's what I mean is membrane and spike specific or spike reactive. I don't go into any depth, but we didn't actually find any different between the profile of membrane and spike reactive CD4 T cells. So we kind of combined everything. But overall, we got good numbers from a lot of the donors. So it was, just over 100,000 viral reactive CD4 T cells, and then we got, we sorted a lot more than that, but over the process of sorting and generating the beads for the 10x for the single cell sequencing, we get over 100,000 viral reactive CD4 T cells. This is a UMAP projection of all the cells that we sequenced. Now, what this kind of shows is all of the, as you get with kind of high-level flow cytometry data, you have all the cells that have a similar transcriptome clustered together. So all the cells in here have a similar kind of transcriptomic profile. And so we can see here that we have kind of broadly a few different subsets that we can pick up. We can pick up follicular helper cells, Th1s, Th17s, interferon response, CD4-CTLs, central memory, and kind of some cells that we call cycling cells. So you can pick up a lot of different kind of CD4 T cell biology in these cells. So what we could then do next is we could compare the profile of the CD4 T cells reacting to the different peptide pools. And what we found is we had these clusters here, which we called polyfunctional Th1. So these are your kind of classical interferon gamma, IL-2, TNF alpha producing cells, the ones that everybody you'd look at and go, they're polyfunctional, that's a good response to a viral infection. And we found that these were predominantly flu reactive. So over 70% of the cells in these two clusters came from flu stimulated conditions. And then similarly, we found these clusters at the top here, 0, 5, and 7, these all had a T follicular helper profile. So we had lots of CXCL13, IL-21, POU2AF1, CD200, a lot of the kind of markers that are associated with T follicular helper cells. And then we also, down at the bottom here, had a CD4-CTL cluster. So lots of expression of perforin and granzyme, the classical cytotoxic markers that you get in CD8 T cells. And for both of these subsets, they were heavily enriched for SARS-CoV-2 reactive CD4 T cells. So this already kind of shows you that using this technology, you can pull apart, again, kind of two different viral reactivities. Obviously, in this context, you have to be careful because we've taken COVID-19 patients who are probably more having an acute disease, and whereas the flu reactive cells have come from healthy donors. So you can't make a direct comparison between an acute and a kind of memory phase, but this was already relatively interesting that they'd split off quite nicely. We can then use gene sets, which have been kind of published in the literature, to show that actually the different populations that we have are indeed what we thought they were. So this population at the top here of SARS-CoV-2 reactive cells, we could confirm that it was indeed a T follicular helper type signature. And then at the bottom here, similarly, the cytotoxic CD4 T cells, we could see that that was, had quite a high gene signature score here at the bottom as well. And like I mentioned before, we also had these kind of central memory interferon response Th17 and cycling subsets, but I don't talk about those too much because that was kind of evenly split between a lot of the viruses that we tested, and there didn't seem to be anything that was specific for one given virus. Thankfully, so we had this data, and it was quite nice that a few months after we published, the people who'd developed the assay also published some single cell RNA sequencing that they'd done using this assay. So the people who developed the assay published this data set, and they found some very similar results to what we found, to what we had. So they found some TFH like cells and some cytotoxic Th1 cells and some cycling cells. So it was kind of nice to have the findings we had corroborated by a separate group and a separate cohort of patients, different people handling and all that sort of thing. So that was, nice to see that what we found wasn't just completely due to our samples or our methods. We then went on to kind of do some analysis on what actually kind of differentiated between the 40 patients that we had. And from this heat map, what you can broadly see is that we have kind of three groups. We have a cluster here at the top, which is people with a high frequency, a high proportion of TFH cells from one of the clusters, which is cluster 0. We then have a group here which have a high proportion of cells from another TFH cluster, but this is cluster 5. And then we have another group at the end here, which has a high proportion cells which come from the cytotoxic CD4 T cell clusters. So these are the three kind of different groups we had, and as you can sort of see here, this group at the top here with a lot of cells from this cluster 0 are predominantly non-hospitalized, as indicated by the green, whereas everything to the left here in these clusters here, in this cluster here, there are a lot of hospitalized individuals. So as I mentioned, we have this TFH grouping up here, which we split into three clusters based on the parameters that we set. And if we looked at the total proportion of T follicular helper cells in hospitalized and non-hospitalized donors, there didn't appear to be any difference. It was kind of, broadly the same. However, if we split that off and looked at what happens in cluster 5, we could see that there were a lot more, in hospitalized donors, there were a lot more cells in this cluster 5. And conversely, there were a lot more cells in non-hospitalized donors in this cluster 0. So these two clusters here were clearly, it's like a kind of dichotomy. We had hospitalized people who had cells in cluster 5, non-hospitalized people who had TFH cells in cluster 0. So we wanted to kind of figure out what is the difference between these two clusters, because they both come up in our kind of fairly superficial analysis as T follicular helper cells. So we perform what we call single cell differential gene expression analysis. So this does just a direct comparison between the two clusters to kind of pick out what genes are upregulated in one cluster versus another. So this is cluster 5 versus cluster 0. The nomenclature is a bit confusing because what this means is what is upregulated in cluster 5 versus what is downregulated in cluster 5. So what we can see is that upregulated in cluster 5, we have a lot of negative regulators of T cell activation like CD70 and DUSP4. And we have a lot of inhibitory markers as well. So your classic kind of PD1, LAG3, TIM3, these kind of things that, a lot of people talk about in the cancer setting were also highly expressed in this cluster 5. And then upregulated in cluster 0 were, some more classical effector molecules, some IL-2, some TNF-alpha. So really showing that there was quite a strong difference between these two clusters, even though we'd labeled them both as T follicular helper clusters. But perhaps the most striking thing that we found is that actually cluster 5 expressed high levels of perforin and granzyme B. So in this cluster 5, over 60% of the cells express either one or both of these cytotoxic molecules, whereas in cluster 0, around 85% didn't express either. So this was kind of like one of the main findings that we had, and it was reminiscent of a paper that was published by a fellow lab in the Institute, in Shane Crotty's lab in the Institute, where they described this kind of cytotoxic TFH cells, which were associated with, recurrent tonsillitis and worse disease pathogenesis. So this was quite an interesting finding that we found only in hospitalized patients, and something that I've omitted from this slide is that the higher proportion of cells from cluster 5 that we got, the lower the antibody titers in these donors. So there was a clear relationship between cytotoxic TFH and low antibody titers. So I'm going to move on to the kind of CD4 CTL aspect of what we found. So like I said, when I joined the lab, my main goal was to determine the phenotype and functional properties of these pathogen-specific and tissue-specific CD4 CTLs. So I come from a lab in Birmingham, England. I'd done my PhD in an Epstein-Barr virus lab. And what we'd done during that time is actually show that during acute infectious mononucleosis, so that's primary EBV infection, you do get this kind of big population of perforin and granzyme B positive CD4 T cells that arise quickly during disease onset, but during convalescence, which can be roughly described as a week or two later, this population disappears very rapidly. And we used class 2 tetramers to look at the kind of the individual viral reactivities. And we could see that actually in these EBV specific CD4 T cells, they expressed a lot of perforin and granzyme. And that kind of pushed up the overall frequency of CD4 CTLs in the total CD4 T cell population. So I always have had an interest in CD4 CTLs. And unfortunately, when we looked at the kind of frequencies between kind of non-hospitalized and hospitalized, there wasn't a big difference. There were it kind of seemed to be a kind of on or off. You had some donors which had high frequencies of CD4-CTLs, and then we had some non-hospitalized donors as well that also had high frequencies. So there wasn't a clear kind of relationship between CD4-CTLs and pathogenesis or protection. But what we were able to do is kind of look into a bit more detail of what these CD4-CTLs look like at the RNA level, because I'd only looked at protein expression of perforin and granzyme B, whereas here, using single cell technologies, we can really dive into it a bit more. So as you can see here in this cluster at the bottom, we found that actually they were highly enriched for transcripts encoding for multiple chemokines, be they CCL3, 4, and 5, but also for XCL1 and XCL2, which are two chemokines that recruit dendritic cells. So this was quite an interesting point because, again, like I just mentioned, we don't know if these CD4-CTLs can act in a protective manner or if they can be, contribute to pathogenesis, because if they can recruit dendritic cells or any two class 2 expressing cells, then potentially they can also kill them directly via use of perforin and granzyme. So that's kind of one of the aspects that was interesting to see, but we still need to investigate a bit further. So that was all the data I've shown you so far. That was all done at 6-hour stimulation. We didn't isolate CD137 expressing cells at 6 hours. We left the cells for 24 hours, and then we isolated them based on dual expression of CD137 and CD69, as you can see from these plots here. So what that means is that we picked up for T cells and also regulatory T cells, because something I should have explained is that what happens when you stimulate cells is they will upregulate CD137 first, and then they will start to upregulate other activation markers. So eventually CD137 cells, expressing cells, will also co-express CD40 ligand, or in this instance CD69. So you get regulatory T cells, and then also cells which have a more central memory phenotype, which don't get activated as quickly as effector T cells. So we're able to analyze another 40,000 SARS-CoV-2 reactive CD4 T cells using this approach. And broadly speaking, they could be split into two kind of categories. We had a big regulatory T cell population, which was to be expected in this bottom left-hand side. And then we also had a CD4-CTL population, another population of CD4-CTLs that popped up after 24-hour stimulation. So when we compared the two, actually the SARS-CoV-2 reactive Tregs were reduced in hospitalized patients. And even though this looks good, it wasn't statistically significant, but the CD4-CTLs were increased as well in hospitalized patients. So we had kind of this kind of dichotomy of either you have a big Treg response or you have a big CD4-CTL response. And if we plot the graph, there's a clear negative correlation between regulatory T cells and CD4-CTLs. But the finding that there were reduced regulatory T cells in hospitalized patients is perhaps not surprising, because we have, the disease pathogenesis is kind of the immune response, an overactive immune response. So if you do not have the regulatory T cells to dampen that down, then that can probably lead to complications. But we were also interested in looking at, given the fact that we'd found this kind of cytotoxic TFH population, we're also interested in looking at T follicular regulatory CD4 T cells. This is, I guess, a relatively novel kind of population. This is a population that was shown to inhibit the development of granzyme B expressing follicular helper T cells. So the kind of follicular T cells that we found in our hospitalized patients. So as you can see here, if you have these TFR cells, then they will inhibit cytotoxic TFH cells that can produce granzyme B, and then therefore can inhibit antibody secreting cells from doing their job, if you will. And we've also recently in our lab, Simon has just had his paper accepted looking at TFR cells which inhibit anti-tumor immunity and are responsive to immune checkpoint blockade. So I think this is, quite an interesting subset to keep an eye on. And so what we did is we used IL-1R2, which is what Simon kind of uses in his paper as a marker for T follicular regulatory CD4 T cells. And we found that there was expression of this IL-1R2 in the kind of regulatory T cell population. And also, again, that actually the expression of IL-1R2 was greatly increased in non-hospitalized patients compared to hospitalized patients. And we did do a kind of a correlation between the expression, of TFR cells and cluster 5. So this was our cytotoxic TFH cluster. The data, there was a negative trend. It wasn't significant, but it does suggest perhaps that there is a link between the presence of these T follicular regulatory T cells and the kind of cytotoxic TFH population that we found earlier. So if you have a lot of these TFR cells, then you won't get as many of this cytotoxic TFH population, which means you will have better antibody responses, which is what we observed. So I'm just going to briefly touch upon one of the other aspects of using the 10x platform, and that is you can get paired gene expression TCR analysis. So we can plot the clonotype information that we have directly onto the gene expression UMAP. And what we could see from that is that actually you have, a lot of clonal expansion in this T follicular helper region up here, but where we saw the most of it was in this area here in the cytotoxic CD4 T cell clusters, and also in this cluster 11 here, which wasn't on this kind of CD4 CTL island. But when we do some trajectory analysis, we can see that actually there is a link between this cluster 11 and the cluster of CD4 CTLs that we see, indicating that there is a link between the two. So while these cells do not have full kind of perforin granzyme B, NKG7 expression, they do express some of the chemokines. So, but whether that's like a kind of before or after subset, we won't be able to tell from this data. And this is a bit of a big slide, but effectively what you want to look at is that the kind of sharing that you can see between clusters. This is another aspect of doing this kind of high resolution biology, is you can see that the plasticity of CD4 T cells is a very real thing. There's no, putting CD4 T cells into a box and calling them a TH1. There is a lot of sharing between a lot of these clusters that we have here. And a lot of the bigger kind of cells that are shared are actually in this CD4 CTL kind of area here between 6, 9, and 11. So again, that's just another aspect of single cell transcriptomics that is very, very useful. Similarly, at the 24-hour mark, we had big clonal expansion of the CD4 CTLs, but not a huge amount in the regulatory T cell subset. So that was kind of a very brief and quick overview of the work we published last year. So it was kind of just 100,000 SARS-CoV-2 reactive CD4 T cells and this kind of imbalance between cytotoxic T cells, be they TFH or kind of classical CD4 CTL, and then the antibody titers that we saw. So I'll just take a few minutes just to briefly talk over some of the work we're hoping to publish in the next couple of months, and that is kind of the follow-up to this. So we've been able to collect samples, taken about 120 days post-symptom onset from the same cohort that we had before, and look at the reactivity to SARS-CoV-2 now and see what it looks like. And while we're doing this, we're going to assess the flu reactive profile as well. So this then will have a direct comparison of two viruses in the same donors to be able to say, well, this is what a SARS-CoV-2 memory response looks like, and this is what a flu memory response looks like. So we managed to get the follow-up PBMCs from 31 of the donors, and we sequenced just under 43,000 cells, about a 2-to-1 ratio between SARS-CoV-2 and flu. And one of the first interesting observations was that actually the response to SARS-CoV-2 is relatively stable. Shane Crotty at our institute also published a paper showing that 8 months post-infection, you could still find T cells, and we were able to kind of, this is only 4 months, but we replicated that, finding that actually the number of SARS-CoV-2 reactive CD4 T cells we get per million PBMCs remains relatively stable, and in some hospitalized donors it goes up although whether that's due to an increased frequency of T cells in the circulation is we need to check that. But broadly speaking, this is the UMAP of just the 40,000 cells. We still find a TFH population, which is enriched for SARS-CoV-2 reactivity. We have this Th1 multifunctional effector subset enriched for flu reactivity. So again, this is in the same donors. We can kind of find again what we saw earlier on, which is that flu reactive cells have more of an interferon gamma IL-2 TNF alpha producing a profile. And then we still have this CD4 CTL population. And this one was present for both viral conditions. So this was perhaps one of the more interesting findings, is that, from my work with EBV, I assumed that the CD4 CTL population would vanish quite rapidly. But it was interesting to see that this population was still stable even 4 months after infection. And this is just some of the data showing the kind of difference between the SARS-CoV-2 and the flu reactive populations. So the TFH is very much a SARS-CoV-2 kind of profile, and that we were able to confirm by protein expression of CXCR5, so that you can use that as a circulating TFH marker. And also to just show that actually between the two time points that we have, the acute time point and the follow-up, the proportion of TFH as a total of the responding cells remains relatively stable. So it really shows that the immune response, or the T cell response to SARS-CoV-2, does not disappear after 4 months, and it maintains a very similar profile as to what we saw at the beginning of disease. And just quickly on the kind of 24-hour stimulation, this was interesting because we found that, again, we found a very large Treg population, high expression of FOXP3. And then when we looked at the proportion of regulatory T cells, it was actually balanced out again, where we didn't see, a higher proportion in hospitalized. It was actually, there was no statistically significant difference between non-hospitalized and hospitalized. And when we look at the individual cohorts, we see that there's actually no change in the non-hospitalized group, but it's actually an increase in the hospitalized donors, an increase of Tregs in hospitalized donors that appears to have kind of redressed the balance between of Treg cells in non-hospitalized and hospitalized patients. So that's just briefly some of the data that we're working on right now. Some of the future work that we'll look at is we're going to receive 1-year follow-up samples. So this will kind of provide another layer of how stable the response to SARS-CoV-2 is and what it actually looks like at the transcriptomic level. The CD4 CTL story is the part that we're really interested in and we're really diving into right now. So we've got some very interesting data coming out from that hopefully will kind of be interesting for people to read. And also we're going to do some more work on the CD8 T cell response to SARS-CoV-2. I haven't spoken about any of it today, but my colleague Anthony published this paper beginning of this year doing a very similar approach, looking at CD8 T cells in COVID-19 patients. So there will be some follow-up work to that as well to kind of see what the long-term implications of CD8 T cells is. And with that, I'd just like to thank everybody who helped out, from Anthony, Bioinformatics, which did an incredible job to help get us all the data, the sequencing core who helped with all the 10x libraries, the peptide pools, and the flow cytometry core who worked long hours to get all the sorting done. And everyone for listening, and I'll be happy to take some questions.
Dr. Mekif, thank you so much. Beautiful work. So we're up for questions. If people want to raise their hands, then you can ask directly, or if you need translation, I can help out.
So I see there's a question, what kind of stimulation did you use for CD4 T cells? So we did 6-hour stimulation. So either 6 or 24-hour stimulation. That's the kind of standard we use. For the SARS-CoV-2 reactivity, we used overlapping peptide pools from Miltenyi. And then for the flu reactivity, we used peptide pools, which were shared by Alessandro Sette's lab. And these were peptide pools that had more predicted epitopes based on HLA typing rather than overlapping peptide pools.
Great. Well, while people heat up with their questions, I have a question. I thought that was really remarkable that you have a good correlation between the antibody titers in hospitalized patients and the CD4 cytotoxic T cells, but not in the non-hospitalized patients. How do you explain that?
I mean, this is one of those things with transcriptome. It throws out all these intriguing possibilities, and you know, in simplified world, it would be that you have cytotoxic TFH, which don't allow the germinal center to get the B cell maturation going, and then you don't get the antibody production. I mean, that would be the most kind of I guess, straightforward way to explain the correlation or the negative correlation we see between the cytotoxic TFH and antibody titers. But I mean, obviously, you know, it requires a lot more work to do, which we're not working on right now, if I'm honest. It's kind of, but yeah, that would be the simplified explanation, I think, of why the cytotoxic TFH are kind of causing lower antibody titers. But as to why we see them in hospitalized patients and not in non-hospitalized, it's tough to tell. I don't know if it's, you know, comorbidities or anything like that that people have in their immune system that would make them susceptible to development of these cytotoxic TFH. That's something that would I don't really have an answer for, unfortunately.
Yes, that seems to be a very interesting question, but the persistence in your new work, the persistence of the different CD4 response to the different viruses is really astonishing. I mean, each virus has its own response.
Exactly, and this is the great thing about, you know, single cell technology. You, like I said, you don't just look at production of interferon gamma as a marker of what a T cell is doing. You know, there's so much more to it. There's so much more effector functions that they appear to do that we, you know, up until now it's just been difficult to look at. And you know, the stability of the T cell response to SARS-CoV-2 is obviously great news for everyone. But you know, it was very interesting to see. And so I didn't mention this, but the cytotoxic TFH populations disappear as well. They're not stable. They don't. The SARS-CoV-2 reactive cytotoxic TFH populations are absent 4 months after you know, symptom onset. So you know, they're a very kind of transient population. They're not a stable one. But you know, the overall TFH response to SARS-CoV-2 appears very stable. So.
Very interesting. Any recommendations, practical recommendations for people that are going to start with these methods? How would you go about starting? How difficult is it? I mean, I've read the data that 10x provides, and it doesn't seem that complicated. I mean, you buy the little machine. What is the learning curve for these type of assays?
So from a practical standpoint, I think doing the assay and the library construction part, which is the construction of your gene expression, your site-seq and TCR libraries, that's a relatively simple process, I would say. I'm sure Anya will be able to kind of persuade you of that as well. I mean, the difficulty I would argue is the analysis part, because I know 10x has some of their own software that they help, but you know, it was a learning curve for us as a lab. I think the fact that we'd done it before was what really helped us, because we already had, you know, a lot of the kind of scripts and stuff. I mean, Vicente, who's here, will be able to answer it better than I can, but you know, I would say that was probably you know, that's the kind of tough part. But you know, we were very fortunate to have very talented bioinformaticians help us with that. But from a practical standpoint of doing the assay and then doing the kind of GEM, so the cell barcoding that helps you for 10x, and doing the library preparation, I wouldn't say it's a particularly challenging protocol. It's just a lot of PCLs and purifications.
Mm-hmm. Vicente, would you like to comment about the analysis? How difficult is it? Once you get all this data, aren't you just overwhelmed with data? What is the learning curve for analyzing this type of data?
Well, I think it's the same as I mentioned. At first, it's kind of complicated, just because it's definitely overwhelming to have just a lot of data to analyze. But however, once you start with the data set, I would say that it gets more easier and easier. And of note, I think 10x provides a lot of help, and actually the very first steps of the analysis are very simplified with the use of that software. So yeah, to be honest, the very first steps are easy just thanks to that software. But then if you want to go through more in-depth analysis, you definitely need to go to use custom scripts. And that's the most difficult part, I would say, once you learn to use 10x software. But that's also like the challenge itself is also exciting, right? You want to answer questions, so you gotta keep on asking questions about your own data set, and then yeah, you can answer based on those scripts.
And I would also echo that, you know, all the scripts that we use, those are publicly available. We don't, you know, everything we've done is, you know, out there for people to use. We're not hiding anything, because that's, you know, the idea is people can be able to take a lot of the scripts that we've done and just be able to use them themselves. So all that data is available out there for everyone to kind of look at and use as well.
We have one technical question here. Why did you choose 10x and not other techniques like DropSec or the SST platform? I don't really know those.
I think I mean, the choice of 10x was just that it would have been something that had been used in our lab quite a lot. I mean, the thing is, the volume of cells that we were doing, you can't do a plate-based assay, because that's just, you know, the amount of work that goes into doing 100,000 cells. You know, even if you manage to get a 384 well plate going is just, you know, the 10x platform is simplified. Everything's in one tube per library. You know, it's just very good if you want to do, you know, some of the, you know, that there are some, you know, if you want to do SmartSeq or something like that, which is a 96 well based assay, single cell assay, you know, there are some things which might be slightly better, but in terms of, you know, ease of use and doing this many cells, then, you know, this was just the obvious choice for us. I don't work for 10x, by the way, just doesn't sound.
Yes. Thank you very much. Well, I don't know, is any more hot questions?
Somebody else, do you think this different population are influenced by inflammatory microenvironment?
Oh, yes.
It's a very good question. We didn't do any sort of serum assays looking at inflammatory cytokines or things like that. We didn't have that data to hand. It would be something interesting to look at. You know, obviously we're looking at blood, and that's just kind of, we hope that that's a reflection of what's happening in lymph nodes, for instance. But, you know, yeah, getting some more data on that would have been very good as well.
Well, Dr. Mekif, thank you so much. And
no problem.
Now, we'll switch to our next speaker, Dr. Adriana Suarez Gonzalez, who's a Science and Technology Advisor at 10x. So, Adriana, please, you have the floor. Let me see. Let me get you started.
Yeah. Okay.
Okay. Let me. I have to reclaim the
host.
The host. Yeah. Let me get that. Oops. So, claim the host. What happened to the host? Where did the host go? Save chat. Yes. Reclaim host, and Adriana is the host now. Adriana, thank you so much for being with us.
Thank you for the invitation. It's a pleasure being in Colombia virtually. I'm originally from Bucaramanga, so it's always nice to participate on events in the region. I hope you can see my screen now. Yes?
Yeah.
Yes?
Yes.
Perfect. Thank you. It's a hard act to follow after Ben's presentation. And yeah, I appreciate the overview of the work and also the candid comments about using the technology. For my section, I'm going to focus on a general overview of the technology, give you a glimpse of the different solutions, and also give you other examples in immunology. Of course, we need to talk about COVID because there are so many papers coming up using single cell in so many different ways. So we will be talking about COVID, of course. I want really today to show you all these different solutions in the context of immunology, because as you know, and you have been working in this field for a while, it's a field that is full of black boxes that we don't fully understand, but we know that it has a lot of very useful information to understand pathology and human health. So to start opening these black boxes, we really need tools that allow us to have precision, but also depth, and that we are able to scale. So we can really look at a lot of cells, but also a lot of different samples. So I hope that today, after these talks, you go out with this question and maybe these thoughts on how you could potentially use these tools to address your specific scientific questions. We have more than 2,500 publications now using the technology, which is huge. And it is also a source for inspiration to really show us all these different options that we have and opportunities that we have with the technology. Not only that, it can also help us to get inspired, but also it's a great resource for things like data analysis, right? We know that there are so many different ways that we can use to explore these data sets. So I think these bioinformatic tools that are mainly open source are amazingly useful for a lot of our customers. So these publications really bring that options for data analysis, but also for sample preparation, which could be also very specific for each experiment. So at 10x, we have a number of different tools. We are going beyond bulk analysis. So bulk has been extremely useful, but it has been also very limited in terms of how we can address the complexity of biology with bulk measurements. So at 10x, we have two different platforms, the single cell that we heard before. And here we're actually getting measurements from each cell, and we can measure different modalities from the cells. So we saw the examples with gene expression, also with T-cell receptor sequencing. We can also look at cell surface proteins, and even look at open regions of chromatin for epigenetics. And we could do some of these measurements simultaneously from the same cell. On the spatial aspect, we are working heavily on expanding that platform. So we call it Visium. And here we can look at these gene expression profiles and also proteomics from the tissues directly. And I'm gonna give you a glimpse of that and how this could be used in immunology at the end of the talk. So when you get into the world of 10x, you're going to have a lot of different options in terms of the type of tools that you can use for your scientific research. So there's a lot of conversations that you might have with 10x, or in Colombia, if you're working in Colombia, we are working with ROChem distributors. So they are going to be your gate to the 10x resources. And so we can have conversations about experimental design, about sample preparation, about even data analysis options for your experiments. So I think this is extremely useful when you start this journey with single cell or spatial analysis. When we talk about single cell, this is going to be your friend. So this is the Chromium Instrument. It's a versatile machine that allows you to run all these different assays that I have been mentioning. So gene expression, immune profiling, single cell proteomics. Is very small. We actually, at the beginning of the development of this product, it was called Toaster because of the small footprint that it has. But it's really powerful because it allows you to get into the single cell analysis at scale. So Anya is going to work in more detail about how this works, but I just want to show you basically the secret sauce of this technology is the capacity that we have to isolate cells and barcode molecules of interest. So we have a lot of flexibility in terms of the type of molecules we can target and the barcodes we use to target those cells. Those barcodes are basically the cellular address of those molecules that allows to identify where those molecules came from. Another thing that you see here is that the input that we use is very flexible as well. So we can work with cells or nuclei, and we can label those cells in different ways to capture specific pieces of information, like, for example, cell sulfate proteins or even antigen specificity. So there are different ways on how you can perform these experiments to get the most out of it based on your scientific questions. And when we think about immunology, of course, we have a lot of opportunities here because we have so many different types of cells that we can start looking at different modalities within the same cell. So if you're looking at T cells or B cells, we have your gene expression, you have your cell sulfate markers. We also have the receptors in the cells. And then we can also look even at antigen specificity. And for other types of cells in the innate system, we will have the possibility of measured gene expression and cell sulfate proteins. So I'm going to give you a tour of examples on how this is being used in different contexts. And I'm going to start with measuring this modality of gene expression in single cell. So this example, it's of course related to COVID, but from the vaccine front. So this was done with the Moderna vaccine, the mRNA 1273 vaccine. And this was basically the preclinical trials that were done with hamsters models. So this drove some of the clinical trials that moved into patients by looking at the immune response after vaccination in these animal models and basically selecting one of the best configurations here. So they had different individuals, different groups. They had a naive set of hamsters, and they also had two groups that they infected. One group was a mock vaccinated, and the other group was vaccinated with the mRNA vaccine. And so here we see, and you saw some of that already, on how we can portray this information in different ways. So here we see each is basically a reduction of the dimensions of this data set, so we can visualize it in a 2D space. And each dot represents a cell. This is all the data set together. So we are just looking at the type of cells that we can recover. And this was done with lung tissue from these hamsters. So we see the presence of natural killer cells, different types of T cells, B cells, and even endothelial cells that we can explore here as we were working with lung tissue. But of course, what we want to see is the differences between all these different groups of animals that were subjected to vaccination or to infection. So some of the findings in this study where they increase in certain cell populations in the mock vaccinated individuals that were infected. So we saw increase in macrophage population, granulocytes, and dendritic cells. But we saw a reduction in the response of CD4 T cells and CD8 activated cells. So very interesting findings here on how we see the infiltration of these inflammatory cells in mock infected individuals. But of course, we don't see that in the naive and vaccinated group with mRNA. The other aspect that we can look at is, as we saw before, you get so much information that there's so many options in terms of how you process this data. So you can look at the host response, and then also you can look at the presence of these viral reads in the cells. So in this case, SARS-CoV-2 is it has polyadenylated transcripts, so that's why we are able to capture them in our platform. And so here they noticed that viral transcripts are actually identified in the mock vaccinated individuals, but not in the vaccinated ones that were infected. We also see that the presence of those viral reads are found across different cell types, especially in macrophages and granulocytes. And we can also look at the specific transcripts, viral transcripts that are overexpressed. So in this case, they found ORF1 as the one with the highest hit there. We also can look at different markers in the host associated with immune response. So CCL8 is a chemoattractant for monocytes and macrophages, and it has been shown to be overexpressed during SARS-CoV-2 infections in ferrets. So we see that there is overexpression of this marker, especially in macrophages, and this happens also in vaccine and infected individuals. So it's possible that this is a signal of cellular trafficking opposed to inflammation. So this work shows how vaccination with mRNA impedes SARS-CoV-2 infection. And we see this in lymphoid and in the myeloid compartment, and how we reduce the activation of this viral infection. So we can add another modalities. In this example, we saw the use of single cell gene expression, but we can add the measurement of cell-sulfate proteins to help identify further cell states, for example, in these different cell types. So we know that the more information we have about these cells, the better is going to be our understanding of their phenotype. So this work that I'm going to show here comes from the NIH. And what they wanted to do was to take or basically explore if by looking at these multi-omic assessments of cells, we can predict vaccine response before we vaccinate people. So they took certain samples from volunteers that were working on a clinical trial for influenza and yellow fever vaccination. They took samples from these people before and after vaccination, and then after vaccination, they look at the immune response. So they wanted to see if they could categorize people based on their response to vaccination. So they knew there were high responders and low responders. And then when they went back to the samples before vaccination, they were able to see specific responses associated with vaccine response. And those were mainly in the plasmacytoid dendritic cell compartment and in certain lymphocyte networks. So very interesting data set showing the power of these multi-omic assessments to really make predictions about immune response to vaccination. And then we can add a third modality here. So we saw gene expression combined with protein detection. And so we can add immune profiling by looking at T cell receptors and B cell receptors. So the key here is that we can look at this information in a paired manner. So we can look at the sequences of these receptors from the cell itself. So we can use this to track clonal expansion. And we also saw an example of that. So just to show you a little bit on how that data could look like. So we have the gene expression. We can combine this with cell-free protein detection to identify different cell populations and cell states as we see it here. But then we can look at the specific compartments in the lymphocyte cells. And so if we're looking at your B cells, we can look at the Ig clonotypes and the specific Ig clonotypes in the populations, but also the frequencies of those clones. And the same is true for the TCR clonotypes. So we can look at the behaviors that way. So this is another example in COVID, published recently in Nature Medicine, that took this multi-omic approach. So they had a cohort of different patients with different symptoms, and they compared COVID patients with COVID, and they also had a control. So they took blood samples, they stained their cells with conjugated antibodies, which is basically what we use to capture this protein information from the cells. They ran 10x, and then they got these different modalities from the same cell. They got gene expression, they got T cell receptors and B cell receptor sequences, and then they also got the cell-surface protein information. And this was done for about 800,000 cells. So here we show some of these results. It's a really comprehensive study. So this is just two pieces that I picked up. But here we see the presence of megakaryocyte progenitors in patients or in people that are showing symptoms. And of course, we don't expect to see this in healthy individuals because hematopoiesis doesn't happen in the blood. So this suggests that there are some issues in COVID associated with perturbations of normal functioning of the bone marrow compartment associated with hematopoiesis. So this is the gray bars we see here. And of course, they are absent in healthy and asymptomatic. Another aspect we see in COVID is that it's not only about the severity of the symptoms, but also the duration. And so we talk a lot about long COVID. And so here, this study shows enrichment of a specific cell types when we have for example, short symptoms are associated with enrichment of cells with cytotoxic phenotypes. And then the final piece here is associated with the clonal, the clones that are being expanded during COVID disease. So they saw that specific clones in the CD8 effector T cell compartment was expanded, and it expanded even more in critical patients. So they were able to identify these specific cells. And of course, there's so much information here. So it's a great data set that I think you could mine and then start coming up with hypotheses for your single cell experiment. The reality is that we have a number of these publications coming up with large data sets, not only looking at one set of tissues, but a number of different tissues, creating this cell atlas of COVID. So it's a super exciting time to really mine some of this data and start generating some ideas for experiments, I think. So looking forward to hearing some of your ideas maybe later as you digest some of this information. So one question is like, how do we get to this type of a scale? And at 10x, we are coming up with solutions to help you do that. So one thing is basically bringing this to a number of samples, basically expanding this to patient samples and doing this at scale. So recently we launched a solution so you can multiplex different samples in one run. It's called CellPlex. And again, if you have questions about this, we can have conversations about experimental design and how to bring this to your experiments. And so we have been talking a lot about single cell. I want to finish this with this last 5-10 minutes that we have with the spatial type of context and analysis we can do. So with single cell, we see that it's very powerful to get the single cell information. But when we do that, and if we have tissues, we are disassociating that tissue. So we lose that spatial context that could be very important if we're interested about heterogeneity or even immune infiltration in our tissues. So with Visium, we have a platform where we can look at the morphology of these tissues, and we can overlay gene expression information to look at these molecular programs that are being expressed in the tissues directly. And recently, I think a couple of weeks ago, we launched a solution for FFPE. So this is opening doors to archived samples, but also these samples that are coming from COVID patients. So with this, we have an integrated solution for data analysis. And here we see an example on how that looks like. So you will have the view of your morphology, as we see here, for example, with IF. And then you could bring on top of that the gene expression information. So we see that represented here by spots and different colors, showing different type of clusters based on gene expression data. So here I have a data set that I'm gonna show briefly on breast cancer. So here we see the pathologist annotation, looking at different regions of, for example, ductal in situ carcinoma and immune infiltration in red. And so we can take that pathologist annotation and then overlay the gene expression information to look at different clusters and different gene expression programs based on this molecular data. So as you can see here, we have that pathologist annotation. And then on top of that, we have a lot of heterogeneity within these regions just based on this molecular data. And so we can do different things. We can start looking at specific markers. So here we see markers that are diagnostics for cancer. And so we see that they are going to highlight different regions of these tissues, bringing us closer into understanding the heterogeneity of these samples. And then we can also look at specific markers associated with immune infiltration. I'm sorry that it's not looking in high definition, but I think the image is very heavy. But here what we can start looking at is at specific markers of cells like T-cell markers, macrophage markers, and then look at that infiltration within the regions. Something that I wanted to show is that we can see infiltration, for example, in regions that were noted as ductal in situ carcinoma by the pathologist, but at the molecular level, we see the expression of these markers, suggesting some immune infiltration there as well, which could be very important for diagnostics, of course. So I'm going to leave it there. Thank you so much, and happy to get any questions.
Dr. Suarez Gonzalez, thank you so much for opening us this field in such a broad way. So interesting, so many opportunities. So questions, you can ask in Spanish. People can ask in Spanish right now. Adriana, your toaster looks very, very simple. What about the machine for doing the spatial work, the Visium?
Yeah, so for Visium, you don't need an instrument from 10x. Visium is a set of slides, and those slides are the slides that allow you to capture the morphology with H&E or immunofluorescence, and then you capture the gene expression. Since you are working with slides, with tissues, and we are also doing staining of that tissue section, then what you will need is access to that histology portion and imaging portion. So we will be thinking about microtome if you're working with FFPE, and then basically microscope that allows you to capture the morphology in your tissues. In terms of the molecular aspect, there are certain standard instruments, a thermocycler, qPCR machine for the library preparation portion. But from the next, you basically are getting the kit with the slides and everything you need to basically do the process. We have a checklist that we share with people that are interested with Visium. So we go through every step of the process and then help you figure it out where to do what and which type of instruments you will need access to.
Thank you. Susana, please.
Sí, hola Adriana, gracias por tu conferencia. I will talk in Spanish because it's easier for me. So Adriana, nosotros vamos a comenzar un estudio en donde vamos a tener tejidos de pacientes antes y después del tratamiento. Es posible tener pues la asesoría de ustedes para hacerlo, pero además como una larga discusión, no muy larga, para ver qué tenemos que prever para poder hacer el análisis de la expresión génica diferencial y ver los cambios que ocurren después de la terapia. Eso es posible con ustedes, que nos asesoren, y ustedes ya han tenido experiencia con esos estudios.
Yes. So the question is about an experiment that is being planned, and you have access to samples that or to patient samples that are coming before and after treatment. So the goal of the experiment is to assess the responses after treatment at the gene expression level. And so, yeah, definitely we can have conversations about the experimental design in terms of which type of samples, how the samples are going to be processed. I mean, when we're thinking about experimental design with this experiment, we spend most of the time really thinking about these aspects. How are these samples being collected? How are they being processed? And then we also go through the QC parameters to make sure that the experiment is a success. So with the next, there is a lot of thought that goes into preparing the sample. So we have very nice looking data at the other end. So you want to make your bioinformaticians happy and the people that are analyzing the data happy by making an effort to make a cell suspension that is in excellent quality. The nice thing is that we know these systems so much that we know what are the things that drive quality. So we can tell you we need high viability, we need to know how many cells. And so all these aspects we can discuss, and if there are issues, we can troubleshoot with you too, like what are things that we can change to improve that quality. In terms of data analysis, we do have bioinformatic tools. So that allows you to basically process the data that you get from the sequencer and then get output files that you can visualize in the Loop browser, which is also a tool from the Next. But there are a number of different resources online. So we can also provide guidance on the type of tools that you might need to basically get to your scientific goal. So yeah, happy to have a conversation afterwards and talk more about your experiment.
So thank you, Adriana. Could you send us your mail, please?
Yes, I will post it here.
Thank you.
I think we have a question from Juanita. Juanita, would you like to answer? Is this question for Adriana, or would this question be for Benjamin?
Él ya me contestó en el chat, gracias.
Ah, listo, perfecto. ¿Más preguntas para Adriana? Listo. Adriana, how expanded are these technologies in Latin America? I mean, have you already sold these machines in Colombia or in our neighborhood?
So in the neighborhood, yes. So Brazil, Argentina, we are working with a number of researchers in Chile, Peru. We are still working too, but there are machines in Brazil, Argentina, and Mexico. I think I'm not missing anyone. And yeah, so we have distributors in Colombia and Peru. So yeah, maybe you'll be the first if you
Yes.
start.
Yes, this is an opportunity to thank Roquem Biocare, that's your distributor in Colombia, who has been very helpful in organizing this talk. So thank you. I don't know if people from Roquem are here, but thank you very much for collaborating. Any more questions? Well, if there are no more questions, we might just go on to Dr. Anja Bastian, who is, I forgot your title. I am so sorry. Who
I can help you.
I'm so sorry. Yes, you are
field application scientist.
Field applications. Yes, I'm sorry.
No worries.
Okay, Dr. Bastian, you have the floor.
I think I need to be the host, though, right? Do I need to?
Yeah.
Okay.
Let me get you the host. I've got it here, and you got make host. You are the host. There you go.
Okay. Let me share my screen and switch over my camera. All right. Yeah. So I'm excited to talk to you guys about, kind of how to get started, some best practices after Adriana kind of showed you what you can do with the technologies and gave you a nice overview of the solutions in general. So the agenda for today is just to go over just the single cell gene expression solution and what kind of a little bit more into the biochemistry, a little bit more about the special sauce. And then I'll talk about the best practices and focusing a little bit on the sample preparation. And then if I have time, I'll show you a quick demo from our loop browser, which we use to do some of the single cell analysis. So some of the tools that you can do. And so just to kind of give you an overview of the single cell solutions. So I won't be talking about the spatial that Adriana just touched on, but I'll be talking about the single cell gene expression solutions. So we have basically 4 different types of single cell solutions. One of them is our flagship product, the single cell gene expression solution. We also call that the 3' assay. And then we have the immune profiling. So that's the one that Dr. Metcalf were using. And both of those are profiling the gene expression or the transcriptome. You can also overlay some of that cell surface protein, so using those barcoded antibodies. But then with the single cell immune profiling solution, you can also get that paired T cell receptor, B cell receptor chains there, as well as some antigens. So you can barcode some antigens and pull out cells that have those antigens present. And then the other solutions we have is the single cell attack assay, or where we can profile accessible chromatin. And then one of the newest single cell assays we came out with is combining these two, the single cell gene expression assay with the attack assay. So now we can profile the transcriptome as well as the gene expression parts. So basically DNA and RNA from the same, in this case, we're using nuclei as an input. But in this talk, I'll focus more on kind of the single cell solutions and talk a little bit about the best practices, some of the sample prep handling considerations. But just to give you a big picture overview of the solution. So you input into the machine either cells or nuclei. For the immune profiling one, we will have to have cells. It's not compatible with nuclei. But you can either use cells or nuclei for the gene expression assay. And then we have all the kits to get you to a sequencing ready library. And so the instrument is shown here, as Adriana mentioned. It doesn't take up a lot of space. And then the other instruments you just need are like thermocyclers, pretty standard equipment. And then, we sequence it on an Illumina sequencer. And then we have the data analysis tools, and they're all, free to use. So the Cell Ranger pipeline is the one that's upfront does some of those alignments, that processing of the sequencing data. And then the loop browser is a visualization tool. So that's a screenshot that you see here where you can visualize some of your data. And I'll give you a brief demo of brief introduction of what you can do with it. But a little bit more about what happens actually in the instrument. So Adriana already showed you, right, this graphic here. And this is basically an illustration of what happens in the instrument in these microfluidics chips. So we load gel beads, and that's really the special sauce, as she mentioned. And then we load cells. And here I've added a picture, right? We can label these cells with barcoded antibodies. So instead of using your fluorescent antibody, you can use a barcoded one. And really, and you're not limited by spectral overlap in this case. So the example Adriana showed was the researcher used over 80 different antibodies with no problem with interference, right, or spectral overlap. So that's a really nice add-on feature. So you're not just getting the gene expression, all the mRNA from inside the cell, but also some of that cell surface protein information. But we load the cells, and then they get partitioned into these little droplets with the gel beads that deliver the barcode. And the goal really is to isolate each cell with a barcode. And so that's what you can see here. We have an oil phase, and so we're just partitioning, right, the cells and the gel beads into this oil phase. Then we can collect it, and then the barcoding just happens in a thermocycler. Once everything is barcoded, we basically can make just a bulk library, right? Everything is barcoded, so we can treat it the same way. But 10x provides all the reagents to get you to a sequencing ready library. And then we can, process it, go through all the cell ranger steps, analysis. But one of the outputs is all the cells that you profiled. So here it's up to here's in this case it's 5,000, and then all the genes, and then how many transcripts you have per cell for each gene. So that's one of the outputs that usually the bioinformaticians use because it's a huge file, but we also process that and then give you something you can visualize. So do some of that secondary analysis. But a little bit more on the gel beads, really the secret sauce. So the gel beads deliver the 10x barcode or the cell address. So that's shown right here. So we have these little oligos, they function as primers. So they have a poly-DT. So we capture polyadenylated mRNA. And so that's one of the requirements. But basically poly-DT captures the polyadenylated mRNA. And then we have, we add on the 10x barcode when we go through the reverse transcription. So when we make cDNA. So all the mRNA molecules within the droplet will have the same 10x barcode. And that's basically, right, allows us to identify the cell later on. And that's really the nice advantage, right? Once everything is barcoded, we can put everything in one tube, not like, the 96 well plates where you kind of have to keep things separated, but we can pull everything together and make one library. And then we have a UMI here that's for transcript counting. So that's also allows us to address any, PCR duplication and just really count individual transcripts, because every cDNA molecule we make will get a unique, just a random 12 base pair sequence here. So that's also a really nice feature, allows us really to count, individual transcripts. And then the other oligos you see on here that I have kind of grayed out. So let me go over to this one, has this capture sequence on it. So instead of a poly-DT, which is for capturing polyadenylated mRNA, we have this known sequence, right? Just this 22 base pair capture sequence. And so what we use this for is for our antibodies. And so I'll show you a picture now of the antibody. So you're probably used to using antibodies with, fluorophores. So instead of a fluorophore, we add on these nucleotide sequence. This is just for, Illumina sequencing. We have an antibody barcode that it tells us the ID of the barcode, so of the antibody. So that's why, there's almost no limit on how many antibodies you can pull together as long as they have a unique barcode. And there's really like up to the millions in terms of barcode possibilities. But then we also have this capture sequence, right? And that's the one that's complementary to what's on the gel bead. So if I switch back to the gel bead, you can see here, right, we have that capture sequence. So all the antibody molecules that are captured will also have the same 10x barcode as the mRNA. And this way we can link together the mRNA as well as the antibody information, and get both, right, the cell surface information and the transcriptome information from the same cell. And right now it's only cell surface proteins, but we're working on making intracellular proteins also available. But yeah, the BioLegend is the one that provides these antibodies, and they're just called total-seq. In this case, if we're using the 3' assay, it's total-seq B, and then for the 5' is total-seq C antibodies. And they have some pre-made panels also, but they have otherwise you can also pick your specific, favorite surface proteins, and I think they have around 400 different ones. So likely that your cell surface protein of interest will be one of them. And just to kind of give you an idea of what the workflow entails. So we start off with cells or nuclei, and the preparation can vary a lot, right? Depending on what kind of samples you work with. If it's something straightforward like PBMCs, may only take 30 minutes, right? To maybe do a Fico gradient, and wash your cells, count them, and they're ready to go. But if you're isolating cells from, let's say, a solid tumor, that will take, quite a bit longer. You have to digest the tissue, maybe remove some dead cells, right? So that processing can take, several hours even. But then we go through the gem generation or the droplet generation, basically, which happens in the instrument. And that process is actually only takes 18 minutes, where we generate the droplets, barcoding the thermocycler. But then we have cDNA, we amplify the cDNA, and then we split it into, if we're only doing gene expression, we'll only have this part. But if we have the cell surface protein information as well, we'll have 2 separate libraries that we're constructing. And both of those get sequenced. And then in comparison, I want to show you just the 5' assay. So that's our 5' immune profiling assay. And the 5' is just because that's the part of the mRNA molecule that we're capturing. And for the immune profiling, right, that's where we have the VDJ sequence, right, on that 5' end of the mRNA. And that's why we have those 2 different options. But with the immune profiling, the workflow is basically identical until you get down to, where you're profiling the gene expression, cell surface protein, that's also an option. But really this specific thing that you can do with the 5' immune profiling is that you get those paired T cell and B cell receptors, right? And so that's unique to that 5' solution. But you can get up to, 4 different libraries, actually, if you include the T cell and B cell. So you can get a lot of information, right? The transcriptome, cell surface protein or antigen information too, and then the T cell and B cell if your sample has both of those cells present. And then we sequence them. And so I'll just show you a quick example of what the final library look like for the gene expression libraries. And so the gene expression libraries are just pretty much standard Illumina sequencing libraries. But the difference is, right, we have our 10x barcode in there, and then the UMIs, right? That's again, that cell ID information. But we can sequence them on any Illumina sequencers, really. The MiSeq may be too low throughput to sequence, a significant number of cells, but it can be used for some QC studies or for a very low cell number. And then we recommend 20,000 reads per cell as a minimum sequencing depth. So if you're sequencing 10,000 cells, that's 200 million reads per sample. If you're only doing 1,000, that's 20 million. And so that will kind of dictate what sequencers you're using. And also will depend, how much you want to sequence will also depend on the cell type you're using. And so here I've shown the sequencing depth, so the 20,000 that we kind of recommend as a starting sequencing depth, and then the number of genes you can detect per cell. And then here in the different lines, we have different cell lines or cell types. So starting from the bottom, this is the PBMC sample. And you don't really need to sequence deeper than 20,000 because it starts to kind of saturate, right? And so that's why for PBMCs, 20,000 is great. If you go a little bit deeper, you can maybe get a few more genes. And then for primary cells, usually we require a little bit less sequencing. But then for cell lines, right? Those are, large cell lines, have everything turned on, pretty large transcriptome. So we're actually getting more genes per cell. So it's really cell dependent, cell type dependent, how many genes per cell you're getting, and also what the sequencing depth will be to get the same number of genes per cell. Because here you could technically, right, for the cell line sequence deeper and get more genes, but 4,000 genes is already a lot of data points, right, to use. Even 1,000 or 2,000, right, is a lot of information about each individual cell. And so, but this is just to give you an idea that the complexity between different cells will vary. But 20,000 gives us a good starting sequencing depth. But then, so a little bit about what the chip looks like. So I mentioned we combine cells with these gel beads, and then we have this oil well or this recovery well. And so we combine the fluids flow through the chip and it gets combined in this well up here. So you can process up to 8 samples at once. That might be a little bit challenging to get everything ready, but the chip is actually not the expensive component. So it's more the reagents that you load. So a lot of people load 1 to 4 samples. But what I wanted to highlight here is that we can, per channel, we can profile 500 to 10,000 cells. So technically you can do up to 80,000 per chip, but that would be 8 reactions. And so we can do anywhere from 500 to 10,000. But now the next question is, how many cells should I profile? And so here I have a little example of if you have a fixed sequencing budget, right? So on our end, it doesn't matter if you're doing 500 cells or 10,000 cells, the cost won't change. But if you have a kind of fixed sequencing budget, you can either profile fewer cells and use a lot of reads, or distribute those same number of reads across many cells. So you can see, right, the different shades of red here. And so then you have some cells will cluster together, but usually the more cells you have, the better the clustering will be, right? So sometimes you have only very few cells, they won't form as nice clusters or subclusters. So that'd be an advantage of profiling more cells. And you can already at lower sequencing depths be able to see some subtypes as well. But those are just some of the considerations when thinking about how many cells to start with. And so in the next section, I'll talk about the samples that we put into the system. So I'll talk a little bit about the sample preparation recommendations. And so that's a really critical part, as Adriana already kind of mentioned in the Q&A, right? We want to put high quality samples in to get high quality data out. And so we have, a sample prep team at 10x that focuses just on this, coming up with protocols that help you get started. And me and her are there to also assist you with getting started with the sample preparation discussion. But we have several resources on our website. So some general protocols, optimal sample preparation, cell counting, how to isolate nuclei. We also have some that are more tissue specific. So for working with PBMCs or mouse embryonic brain, or like tumor dissociation protocol as well. We don't have protocols for all kinds of tissues. There's just so many different tissue types out there, but we have at least some starting points and, can point you into the right direction if you have a very specific tissue type that you want to dissociate into a single cell suspension. And so we also have some other more specific protocols for like CD3 enrichment using some of the bead-based methods or removal of dead cells. So next one I highlight this general cell preparation guide. And so that's the one shown here. And it's just a great starting point if you're new to single cell sequencing. It tells you how to kind of wash your cells, count them, how to prepare them. So I'll go into quickly some of the best practices and recommendations of the requirements for your cells. And so obviously when we're working with single cell gene expression studies, we need to make sure we have a single cell suspension. But we also need it to be free of aggregates, right? Cell clumps, shouldn't have a lot of dead cells. And then, not a lot of just mRNA floating around or inhibitors. So of the RT reaction, so they'll be like EDTA. But if you have a cell suspension, PBMCs or sorted cells, those are really straightforward to use, right? They're already in a single cell suspension, or if they're cultured, right? They're just you just trypsinize them. But those don't require a whole lot of sample prepping other than just washing, and hopefully the next step, right, is like we want to make sure we have high viability going in. First of all, we don't want to sequence a bunch of dead cells, but also dead cells will cause a lot of background even in the live cells that get captured. And so we want to have at least like 90%, ideally, if you're working with something more straightforward like PBMCs or cell lines. But if you're working with like a primary tissue, 70% viability is also still acceptable there. And yeah, we're here to help you, get to that 70 or 90% when you kind of do some of the optimization. But it's really critical to have high quality samples going in to get the best quality data out of it. And so we recommend handling the cells gently, pipetting slowly, just some precautions that we have to prevent cell lysis. We also recommend to remove kind of aggregates or cell clumps, to use like a cell strainer, because we're working with really tiny microfluidics channels. And so we don't want big aggregates kind of causing a clog in those channels. And then, as Adriana also mentioned, like quantitating your cells before going into the system. So if you want to recover 10,000 cells, you want to make sure you get an accurate cell count beforehand, so you're loading the appropriate number of cells. So if your cell count is off, then the number of cells you get back right after sequencing is going to be off too. So we really stress this accurate cell counting as well. And then once your cells are ready and counted, we want to get them into the instrument quickly, ideally within 30 minutes, and not have them sit on ice for extended periods of time. And so those are just some of our general recommendations, and we can definitely talk about that in more detail when you're planning your experiments and getting started. So we have a couple examples here that I'm showing for the tumor dissociation protocol. For that one, we're using the Gentle Max. So for Miltenyi, it's the product, right, that allows for dissociation of those tumors. But there's also other protocols that are using just some enzymatic cocktails that you can make yourself, and use like a pestle to kind of get rid of some of the tissue and break it up. But there's some other recommendations as well if you don't have the access to the Gentle Max. And then our protocol for the dissociation of mouse embryonic neuronal tissue, we just use a cocktail of papain and pipette up and down with a transfer pipette, right? And then kind of isolate those cells to first break up the tissue, right? And then kind of clean up the cell sample from removing debris, and then counting them, and then they're ready to be loaded. And some sample types are a little bit more challenging to work with than others, but I think our customers have been able to pretty much get everything to work in a way. So definitely a lot of resources. We definitely take advantage of those publications too, those 2,500. But in the last few minutes, I just want to switch gear really quick. So let me stop sharing this screen and share a different one, and show you a quick loop browser demo of what your data would look like when you're, and I think you can see my screen now, right? Let me double check. Yeah, so this is the loop browser software that comes free to download from the website. And I have this example data set, which is just 10,000 PBMCs. And they were labeled with those oligo-tagged antibodies. So we actually have, I can show, I put the list in here. We have, I think 14 different antibodies that we added also, and they were labeled with. So we have, the transcriptome information, but also the antibody information. And so when you first open this, you see this graph-based clustering. So there's an algorithm that generates, it's kind of unbiased, and generates as many clusters as there is heterogeneity in your data set, basically. So there's, about 11 unique populations, subpopulations, or cell states present. And so we can actually, go through and if we go into just the active feature list here, type in CD14, and we have the gene and the antibody information. But I'll just show you the gene for now. And so we see here the gene expression. Very clearly, this cluster would be our, monocytes here, and we can easily select them because, the data set doesn't come with any annotation, right? So it's just going to have the clusters, and then you kind of, can use some of the known markers to annotate what's present. And so another one would be, right, CD3E, right? There probably should be T cells present. It's a PBMC data set. So, right, we can easily identify those. So that's something that we can do pretty easily on some of the more known markers. Oops, what just happened? The beauty of a live demo. So those will be our T cells. And lastly, right, I can type in another marker for our B cells. Right here, right, that's a gene for CD20. So we can also mark those as our cells and B cells. So that would be, right, one of the first steps to kind of see what cell types are there. I just broadly categorize them. There's definitely a lot of cells, subtypes as well present. But basically, right, when you go in the gene expression box here, right, you can type in, right, any genes of interest, like I just did. But we also have the total seq information present. So if we wanted to see PD1, right, and the protein expression. And here, as you know, right, with antibody staining, you sometimes have a lot of background. So I'll change this threshold a little bit, remove some of that background to see more of that positive population here. So you have, right, the whole transcriptome available and those 14 different antibodies markers that the cells were stained with. And with the antibody part is we also can have this nice feature plot. And so this is going to be very similar to what you may see, right, with, if you've done a lot of flow cytometry, you can now type in, right, your antibody markers. So, and get your pretty much a flow plot. And the information we see here is the antibody information, but now we can also look at the gene expression information. So one thing I want to do, right, very simple little study we can do right here really quickly. We can call these maybe, put them in the cell category as well, and call these, CD4 high expressing ones. So save those there. And then I'll name these ones also in the cell category, CD4 low. And let's say, I just wanted to compare those two populations and see what's different on the gene expression level. So down here, we have this, feature type is gene, so gene expression, and then we can click on this little calculator. And it will do differential expression analysis between those two populations that we highlighted. And it's using, again, the whole transcriptome to do this, not just the antibodies that we profiled. And so that's great for discovery if you want to see some new markers. This is a very simple example. I think many of you probably know what the difference is going to be between the CD4 high and CD4 low, but right, you can do that with any markers. And the nice part is you can integrate the gene expression information as well as the antibody information very easily here. And so this is a nice little tool for us. Let me admit just some more people that just came in, but some nice tool that we can do. Yeah, and it takes a little bit, just a few minutes, but you can clearly see that the CD4 high have a lot of T cell expression, right? And T cell markers. And then on the CD4 low, oops, clicked wrong, right? We see CD14, some other, more standard monocyte markers. And that's also very similar. If I click on what I previously identified as monocytes, oops, they should be present there. Yeah. And so this was just a very quick demo just to kind of show you how you can, use your data. It's really, the Loop browser is made for maybe more for the biologist to get you started, get you looking at your data, and to be able to facilitate the communication between the biologist and the bioinformatician And so you can kind of might get started and be live on the same page and do the discovery together. So, but hopefully, yeah, this was gave you a little idea what you can do with some of these data sets and some of the power of the 10x solutions. So with that, I'll go back to Manuel.
Dr. Bastian, thank you so much. I think you got many people interested here in your technology. We are flow cytometrists, as a matter of fact, and this seems like you're really grabbing into the field, no? How? Just a very simple, quick first question: how does flow cytometry compare with this method? I mean, we're having a big effort in getting 24 flow cytometry panels, and now you're saying that you can get 100 with no problem with compensation. How accurate is the technology?
Yeah, and so in terms of compensation,
if you compare a flow cytometry experiment with a 10x TrueSec, how does it compare?
Yeah, it's very comparable, and we've done comparison studies where we did fluorescent antibodies and our antibodies, and it's basically the antibody binds and we capture it, and you can actually quantify exactly how many antibodies were bound. Because of the UMI tag we have, we can say, okay, there was 1,000 antibodies bound to the cell, right? So we can actually put a number to it, and so it's very comparable, and it's a great technology. You still maybe can't fully get away from titrating your antibodies. That's still something you may have to do. And when you have 80 antibodies, that might take a little while. But some people I know don't, including myself, sometimes just use the 0.25 micrograms or something of antibodies, and it usually works. But the best practice is to do still titration. But yeah, we can see histograms, right? Like the negative population, positive population. So really, really powerful solution. Yeah.
What would you replace for a viability dye?
And so we can't see that, right, in our 10x data. That's something we would want to do upfront, right? Sorting with PI negative, PI positive. But we have the whole transcriptome, right? So if we do have dead cells present, first of all, most of the time, all the antibodies are binding to the dead cells. That's one way to identify them. Or we can use the transcriptome information, right? So dead cells will start expressing certain genes, right? Like usually an overrepresentation of mitochondrial genes. And we can, based on the gene expression profile, just remove those out of the data set, right? And so we don't have a propylium iodide or live dead cell stain, but there's other ways to do that.
Thank you so much. More questions?
Oh, mitochondrial analysis. I saw a question come in. So is it possible to do mitochondrial analysis? We do sometimes get mitochondrial genes, which we usually don't want. So it might be kind of too small. That's just if you're just isolating mitochondria and trying to profile those, there would be maybe a challenge with the sensitivity. And Adriana, yeah, maybe if you have, I saw you come on, if you have some other information.
Yeah, so yeah, as Anya mentioned, with the gene expression, usually people filter that out. And we also have the single cell attack solution, which was designed to be used with nuclei to minimize the mitochondrial reads you get. But there has been these papers that use cells with the single cell attack assay to recover the mitochondrial genomes. And so you are capturing the DNA of the mitochondria. So they have shown that you can identify rare clones and do this mitochondrial analysis using the single cell attack with fixed cells. So it's something that we don't officially support at 10x, but there are publications with protocols out there. So if you're interested, we can have, yes, Susanna, we can have discussions about that.
Yes, thank you, Adriana. Thank you.
I think there's another question about formalin fixed.
Yes, please, Adriana.
Anya, do you want to take that or?
Yeah, sure. So I think is that for the single cell, this so yeah, from formalin fixed paraffin embedded tissue, we can't isolate single cells from that. We have a solution coming out where we can fix cells if they're in a single cell suspension or a tissue just with the formalin and be able to process later, but that's something that's coming next year. So we don't have that available. But if you're looking into fixing tissue, it's maybe a cell suspension, we do have a methanol fixation protocol to fix that in place. But getting live cells from FFPE tissue, we're not able to do that. Anything you want to add, Adriana? Yeah.
Could you please clarify? I don't think I understood your answer. You can't do from
so FFPE, right? The formalin fixed paraffin embedded tissue, right, has all this tissue basically embedded in wax. We're not going to be able to get live cells out of tissue like that for processing. So it's good to start either with fresh tissue when you try to profile cells, right, and make that into a single cell suspension, or starting, we also have an option to start with frozen tissue to isolate nuclei. And then sometime next year, we have the option to do formalin fixation and then take those formalin fixed cells and then profile them, but not with the paraffin present.
I got it.
But actually, maybe to add to that too, FFP did come up earlier in Adriana's talk, and that's for our tissue section. So that wouldn't be a single cell profiling. With the FFP, there would be tissue sections, right, like histological sections, and we can do transcriptomic profiling with that. But that wouldn't be the single cell solution that I kind of just talked about.
Got it. That was
okay. I think you're on mute, Mona.
Yes, I'm sorry. More questions? Well, I think we've gone over everything. I think we have learned a great deal, and I would really like to thank all of the three of you for your beautiful talks. This is really a very exciting time. We're learning so much and really having to change our ways of thinking of the immune system, our ways of thinking of doing science. It's really, we have what they call the Chinese curse. You will live in interesting times. We are really have a lot to learn and a lot of work ahead of us. And with this, well, if there are no more questions, I think we can end the meeting. Any of you would like to say anything before we finish? Well, if not, thank you again so much, and we will be recording your presentations, and we will be posting them on the association website. Is this okay with you? Okay, yes. Perfect.
Yeah, yes.
Well, then with that, we will finish. Thank you again so much. Bye-bye to everybody.
Muchas gracias.
Gracias,
Thank
gracias.
Gracias.
Yeah.
Gracias.